Good afternoon. Thank you for coming to the ophthalmological session. We are going to talk about new therapeutic approaches in ophthalmology. As you probably know, during years, drug discovery in ophthalmology has been a kind of Cinderella. So most of the drugs arriving to ophthalmological indications came from a general drug discovery, and someone has reused or has redirected these drugs to ophthalmology. But since some years ago, this paradigm has changed, and now we have a quite productive ophthalmological drug discovery process. And which are the pushers on this change of paradigm? First, that we have new discovery platforms. You know that the telescope of Galileo was an excellent discovery platform for celestial bodies, but has a limited power to discover new celestial bodies. When we change from the telescope of Galileo to new platforms in astronomy, we are able to discover millions of new celestial bodies. In the same way, we had a very nice discovery platform, a very productive discovery platform that is chemistry, but nowadays we have really a platform proliferation, and we have a large number of new possibilities to discover drugs. And if we put our attention in the uptimers, certainly something changed in drug discovery in ophthalmology when in 2014 an ophthalmer report good clinical results in AMD. In addition to new discovery platforms, we have also brand new delivery platforms like intravitreal injections, ocular implants, and ocular iontophoresis. And the third pusher has been the market growth. Has been a huge growth in the ophthalmological drug market. So the global ophthalmological drug market is estimated to grow at annual accumulative rate of 7%. That means to pass from $16 billion to $35 billion in 2022. This is 2.5 times the rate of the overall pharmaceutical industry. So today we are going to review 3 different approaches in ophthalmological drug discovery: the oligonucleotide-based therapies, the cell-based therapies, and the GPCR target therapies. And I'm particularly proud of the speakers we have joined today. They represent the excellence in translational medicine. They perform world-class research, and more importantly, they are certainly good people. Our first speaker is Dr. Ana Isabel Jimenez, who obtained her PhD in biological sciences from the Complutense University and a management development program diploma from the EOI Business School in Madrid. She completed her postdoctoral training in the Department of Molecular Pathology in the National Cancer Research Center. She moved to the biotech field 15 years ago, and in 2006, she was appointed Director of Research and Development at Scilentis. She was promoted to her current position as Chief Operations Officer at Scilentis in 2013, where she has direct executive and scientific responsibilities on teams successfully developing drugs based on RNA interference for different pathologies. She's co-author of over 20 peer-reviewed publications, 50 communications and lectures, co-inventor of 16 international patent applications, and more than 15 funded research programs. She is mentoring and lecturing in many postgraduate programs and initiatives in the field of health. The title of her conference is Oligonucleotide-Based Therapies, Development of and RNAi compounds for the treatment of ocular conditions. Isabel?
First of all, I would like to thank the organizers for this interesting meeting in ophthalmology. It's the first time that in BIOSPAIN there are a meeting focused in ophthalmology, and we are very happy to present the data. The second present. La segunda. For us, it's very exciting to present the ophthalmological project that we are developing in the company based on the RNA interference technology. First of all, I would like to introduce some topic about the oligonucleotide, because the oligonucleotide represent a new class of therapeutic agents, and these molecules don't fit the typical paradigm of the traditional drug because they have a new mechanism of action and there are new molecules based on oligonucleotide. Oligonucleotides are classified in different categories: antisense, aptamer, sRNA, and microRNA. And all of them are characterized by modulation of the gene expression without modifying the genes. Now, 6 oligos have been approved by the FDA and 4 oligos by the EMA. And it's predicted that the oligonucleotide is one of the major therapies in the next future. Regarding to the mechanism of action, in contrast with the traditional drug like a small drug or biological antibodies that this kind of drug bind to the protein, the oligonucleotide bind to the mRNA and produce the degradation of the mRNA. And if we don't have mRNA, it's not possible the production of the protein. And the design of this molecule is totally specific to the target that we want to silence it. In the last time, there are many programs in clinical trials, and now more than 100 oligonucleotides are in different states of clinical trials. Regarding to our company, Sylentis is a pharmaceutical company founded in 2006, belong to the PharmaMar group, and we are focused in the development of RNA drug for ocular diseases. RNA, as previously mentioned, represents a new mechanism of action, and it is a very new approach to silencing genes involved in different pathology. Our clinical strategy is based on topical administration of RNA drug, and we design a specific software, SerFinder, for the design of the RNA drug, because these drugs are designed using bioinformatic tools. So here we have the pipeline of the company. All of our products are focused in RNA interference technology. Tivanisiran is a product for the treatment of dry eye disease. The product is now running a phase 3 clinical trials. Vamosiran is the second product that we have in the company. The product is for the treatment of glaucoma, and the Vamosiran has completed a phase 2 clinical trials. And we have 2 new research lines in the company with different sRNA to block or trying to silence some targets involved in ocular allergy and retinal diseases. In the following slide, I will put the focus in the development that we are carrying out in the company in dry eye syndrome and retinal diseases. Our product for the treatment of dry eye disease, the name is Tivanisiran, is an sRNA that are specific design for the pathology. Dry eye is a multifactorial disease that is characterized by ocular pain, ocular surface inflammation, and tear film alteration. This is the 3 main alteration in the dry eye patient. Our product, Tivanisiran, is a sRNA targeting the transient receptor potential anion channel subfamily 5, member 1, named TRPV1 receptor. And the TRPV1 receptor is involved in the transmission of the pain and increase the pro-inflammatory cytokine release. In our hands, in the preclinical and clinical study, we demonstrated that when we silencing the TRPV1 receptor with tivanisiran, we control the pain because we reduce the pain, we reduce the pro-inflammatory mediators, and of course we control the inflammation, and increase the production of the mucin. So we improve the quality in the preclinical studies. So tivanisiran is a first-in-class new chemical entity with a different mechanism of action based on RNA interference and block a new target, the capsaicin receptor. The product has been complete 2 phase 2 dose-finding clinical trials in patients with dry eye. And in these clinical trials, we observe an improvement of signs and symptoms in the patient, and we selected the most efficacy doses for further development. Now the product is running a phase 3 clinical trials, HELIX trial, in which we want to analyze an improvement of signs and symptoms in a large population of patients, and we expected the results in the first quarter of next year. Regarding to the efficacy in the phase 2 clinical trial, we demonstrated that our product has immediately efficacy with long-lasting effect, and we improve the signs and symptoms in the patients. And the product is very safety. We don't observe any side effect associated to the medication, are very well tolerated by systemic and ocular toleration. And other targets can be silencing, other ocular diseases can be treated with our compound, and we are analyzing other possible diseases, ocular diseases. As a summary of tivanisiran, our compound is a first-in-class treatment for dry eye patients that block the TRPV1 receptor with a new mechanism of action. And tivanisiran could become the first and the only drug for dry eye disease that control pain, inflammation, and mucin secretion. That is the 3 main components that is necessary to improve in patients with dry eye. Tivanisiran was found to be a potent drug for this kind of patients and show a rapid onset action and very good efficacy and no side effect with the medication. Dry eye is a multifactorial disease with high unmet need, and there are a few medicines approved. Only 2 medicines have been approved for the dry eye. And the market products today have long onset action and ocular side effect. So the results of tivanisiran suggested that our product could be beneficial for the patients. Regarding to our second product for retinal diseases, the name of the product is SIL-8001. The indication of this product is A, macular degeneration, AMD. AMD affects between 30 or 50 million people worldwide, and is one of the main causes of vision loss in the elderly population. The current treatment for AMD are intravitreal injection with anti-VEGF, different VEGF antibody, and the patient doesn't cure only, maintain the visual acuity, but they need frequent injections. In this case, our compound is an siRNA also targeting the not regulated carrion repeat protein named NARP. And NARP is a key controller of the retina neovascularization. When we silencing NARP in the retina, we observe a very good correlation with basal regression and decrease the retinal vascular growth. The mechanism of action is quite complicated because participates many very well targets that not and Wnt. SIL-1801 is a first-in-class new chemical entity with a different mechanism of action and to block a new target different than the market drug, that all of the market drugs are in the VEGF pathway. Our compound reached the retina, and we detected the compound into the cells when we applied the product topically. We administer the product in eye drops. And we demonstrated in the preclinical study that the compound reduced the angiogenesis in the gold standard animal model of the macular degeneration in a similar manner than the intravitreal injection of anti-VEGF therapies. The compound is very safe and well tolerated and not induce any immune response. And we are planning to enter in clinic next year. In a summary of the compound for AMD, for macular degeneration, SIL 8001 is a first-in-class, also new chemical entity designed specific to block a new target in the angiogenesis, NARP. Our compound could be the first topical siRNA administration for AMD and control the retinal angiogenesis, at least in the preclinical study, in the same manner that the intravitreal injections. We complete all of the toxicological package, the regulatory toxicological package, and we don't observe any side effect associated to long-lasting treatment in animals models. And we are planning to enter in clinic next year. As a summary, we consider that the eye is a good target for the RNA interference therapy because it's a confined department and therefore enabled to local delivery of siRNA by topical instillation. Our company is put all of the focus of the company is in topical administration because the patient demand more comfortable or administration for chronic disease, like eye diseases are chronic diseases. RNA offers a significant advantage compared with the market drug because we can block new targets involved in different ocular diseases, our compounds produce sustained efficacy and show very safety profile. This is quite important in ocular products because the eye is very sensitive to drug or chemical compound, and it's necessary to have a very safety profile. And the siRNA have very nice tolerance, ocular tolerance. RNA interference technology could be beneficial for the patient suffering ocular diseases and open the possibility to treat to obtain new treatment for covering all of the unmet needs in the ocular diseases. Thank you very much for your attention.
Thank you, Isabel. We can keep the questions to the end of the session. And now, and thank you for fitting with the time perfectly. Now we have the next speaker is Professor José Carlos Pastor. He is full professor of ophthalmology in the University of Valladolid and chairman of ophthalmology in the Hospital Clínico Universitario of Valladolid. He's founder and director during more than 20 years of the highly reputed IOVA, a landmark institution in translational ophthalmology. Currently, he is director of the Retina Group at IOVA and general coordinator of Red, the cooperative research network of the Carlos III Institute of Health. He is co-inventor of 8 patent applications, author of more than 50 publications, 500 communications, and 32 books or chapters. Director of 43 doctoral theses, member of the editorial board of 12 scientific journals, and Professor Pastor has received 33 national and international awards. He is also founder and scientific director of Vision R&D, the first spin-off of the University of Valladolid. The title of his conference is New Perspectives in Retinal Degenerative Diseases. Thank you.
Thank you, Andres, and thank you, all of you, to be here, despite the heart. Well, let me just as an introduction to summarize several points that I consider of interest. First is retinal diseases has a high prevalence and it's increasingly because most of them are related with the aging process. The second is, unfortunately, we have not successful treatment for most of them. The retinal tissue, this is the retinal tissue, these are the retinal pigment epithelial layer and the photoreceptor. Remember, there is part of the central nervous system. Therefore, we believe that the retinal diseases could be treated probably with therapies developed for CNS. And finally, I think the eyeball and the retina could be a very nice model to test some other treatments, such as those developed for CNS. Because this is a closed and relatively isolated system, and it is very easy to deal with. So let me at the beginning to introduce the major retinal diseases. For sure, the most important one is the diabetic retinopathy. This is information coming from the webpage of the American Academy of Ophthalmology. Remember that diabetic retinopathy is the leading cause of blindness in working age in the developed world. And the prevalence in diabetics varies according to the countries. In Spain, it's around 23–24% of them. In some other countries, such as in Germany, it's around 30%, and it goes up to 43% in the United Kingdom. The second disease, for sure, is age macular degeneration. We, the ophthalmologists, classify the disease in 3 stages. The early one is characterized by pigment alteration on the macula and these subretinal deposit yellows that we call drusen. So the presence of this disease varies from 3.5% in people below 60 years old and goes up to 80% in patients over 85 years old. But the most severe cases, that means those with a strong burden on the visual acuity, goes to the 10% of those people over 85 years old. And the bad news is, according to the estimation, probably the number of people is going to be double by the year to 2040. But there are other diseases also, and for example, the retinal detachment, glaucoma, the pathology myopia, which is a real problem today and in the whole world, and finally, those diseases related with degeneration. The most very well known is retinitis pigmentosa. Let me just a brief comment on glaucoma. Glaucoma was considered the effect of the elevated intraocular pressure or on the optic nerve head. But now the ophthalmologists realize that there is a lot of damage on the retinal layer that's been on the retina itself. So this is a very curious picture because this is the prevalence of the diseases in the developed country. So in black, this is diabetic retinopathy, and this small slice is just dedicated to retinitis pigmentosa. Nevertheless, if you look in PubMed, the publication has this distribution. This is funny because, as you can see, the number of papers published in retinitis pigmentosa is very high. And it is probably because these kind of diseases are very exciting for the basic researchers. But I think the research should be orientated toward the market. And this picture is not only to remind that probably the eye is a window for the soul, I don't know, but for sure it's a window for the brain, without any doubt. During the last year, we have developed this strong image analysis. This is the optical coherent tomography. We call that OCT. And it is able to obtain this kind of picture in vivo, in patient, in seconds, without dilatation of the pupil. And this is amazing because you can support the picture of the histology, and you can recognize all the layers of the retina. The retinal pigment epithelial, the photoreceptor, the optic nerve head layer, and so on and so on. But the most important thing is changes in the wavelength of the nerve fiber layers and the so-called macular volume are absolutely related with diseases of the central nervous system, such as multiple sclerosis, Alzheimer's, and Parkinson. Even more, These changes are not only related with the diagnosis of the disease, but also with the progression of the disease. And some researchers are using that to measure the outcome of the treatment for the basic researchers of the central nervous system. Well, coming back to the retina, there are many possibilities. I recommend you this major review published this year on the neuroprotection strategy for retinal diseases. And I'm going to talk very briefly about the so-called advanced therapies. For sure, today, nowadays, is the time of the genetic approach, without any doubt. This is my friend, this is Robert McLaren, this is the chairman of Oxford, and he published a beautiful review in Ophthalmology in 2016 on gene therapy and stem cell transplantation. But the explosion has been Luxturna, because Spark Therapeutics produced the first treatment, genetic treatment, approved by the FDA for a genetic disease. And for sure, is the most expensive treatment in the world. Is 850,000 euros per treatment. Well, this is an adeno-associated viral vector for a mutation. There is a problem with this mutation. This mutation related to the gene RP65 is relatively rare in Europe. It's very common in the United States, but it is very rare in Europe. Nevertheless, 4 days ago, the European Medical Agency has submitted a positive recommendation for accepting this kind of treatment inside of the European Union. Well, let me concentrate on cell therapy, because our institute is dedicated to the cell therapy. Why cell therapy? Because we belong to the Spanish network of cell therapy of the Institute of Health, Carlos III Institute of Health, and we have a network in the region funded by the local government of Castilla y León. So, and apparently, according to this news, this is a relatively nice investment for the companies. They are investing in cell therapy. There are many sources of stem cells for treating macular diseases. Some of them came from the own eye. I mean, you can find stem cells in the limbus or in the ciliary margin, but you can also use from the umbilical cord, induced pluripotential, fetal, from bone marrow, adipose tissue, and so on and so on. And we concentrate the efforts in mesenchymal cells. Why? Especially for this reason, but because of the immunotolerance, which is I mentioned for the treatment of many patients. So mesenchymal cells have been used for retinal diseases in many ways. They have very peculiar characteristics. One is they go directly to the injured retina because they are, in a way, attracted by chemoattractants. That's funny. In a rat, you can inject in the tail, and the cells go directly to the eye if the eye is injured. Second, if you provide the appropriate stimulus, you can differentiate that into mature cells. And finally, and this is the most important, there are like factories or growth factors. And this is a very interesting property that we are using with the stem cells. The group of retina of the Institute is fully concentrated in this layer. This is the retinal pigment epithelial layer. This is a monolayer. It's a relatively simple tissue, but with a variety of roles and important roles in the vision process. And during this year, we have worked differentiation on the retinal pigment epithelium, and so on and so on. But above all, we have tested the capability of the mesenchymal cells of rescuing cells of the retinal pigment epithelium and what happened with the neuroretina. And this is a very simple experiment. You can cultivate retinal pigment epithelial cells, and you can treat them with mitomycin C. And this is toxic for cells. And what happened? All they die in 3 weeks. But if you co-cultivate with mesenchymal cells, you can rescue part of the retinal pigment epithelial cells. As you can see here, just to make the thing simple, green is good, red is bad. You don't treat, you treat the cells 3 weeks afterward, all of them are die. You co-cultivate with mesenchymal cells, approximately 50% of them are still alive 3 weeks after the treatment. But also, we use a neuroretina culture as a tool to estimate what happened with a more complex tissue. And we can work with porcine retina and with human tissues. We are allowed to work with human under a specific condition. This is a human retina. This is a fresh retina, 3 days in culture, 6 days, and 9 days. And we can test drugs in the neuroretinal tissue, as you can see here, analyzing a lot of properties. And finally, in 2013, we released a patent on the use of the several factors added to the supernatant of the mesenchymal cells. But I would like to provide you an example of the translational research. We saw in 2014 this paper, and it was very nice. They say, why we don't try to treat patients, human patients, instead of rat or rabbits? And well, we started to deal with this disease. This is non-arteritic ischemic optic neuropathy. This is a not very common disease, but it is a disaster for the patient for many reasons. This is, in short, is a head infarct of the optic nerve head. And the bad news for the patient is two. In 25%, there is bilateral affectation in the fellow eye. And the second bad news for the patient is, to the moment, there is not any effective treatment, neither for prevention nor for the control of the fellow eye. So we started with an hypothesis. When you suffer a stroke, there is an area of the brain which is completely destroyed by the ischemia. But this area is surrounded by another area named penumbra area. If you act of this area, you can probably rescue neurons. If you rescue neurons, the functional outcome of the patient is going to be better. And this is our hypothesis with the optic nerve head. We are trying to use this pathway, the paracrine, the production of the growth factor, to rescue neurons of the patient with this neuropathy. So we got the approval of this clinical trial 2 years ago, and the project is still going on, and we are going to produce the first scientific production probably in a couple of months. Obviously, the clinical trial is registered in clinicaltrials.gov. But just to end this small, brief talk, let me remind you that there are another important mechanism also related with the retinal degeneration: eye excitotoxicity, the oxidative stress, inflammation, and some others. And this is another example. So we are working with an important winery in Valladolid. This is not strange. We are close to Ribera de Duero. So these people has a patent on polyphenol. And we are analyzing the antioxidant, anti-inflammatory, and anti-angiogenic properties on a very specific polyphenol of Mata romera. Well, let me, because there are only 2 more slides, and I think the eye is important. I think Andres mentioned that reprofiling, retasking, or whatever you mentioned, or or whatever you name that, this is important. And we believe that in the near future, some drugs developed for the central nervous system are going to be transferred to the eye. I'm more than sure. And don't forget that the eye offers many opportunities to treat by different routes. So not only topical application, but subconjunctival, intravitreal, scleral, and even subretinal. This is the summary. I think retinal diseases market has a very, very nice perspectives because the population is aging, including myself. Second, I think the advanced therapies now show realistic results. Retina is we believe this is a very nice candidate for reprofiling. Drugs developed for CNS, sure, are going to be transferred into the retina and optic nerve diseases. And finally, the eyeball is a privileged place for testing drugs in the central nervous system diseases. And well, let me express my gratitude to the members of the IOBA, the Eye Institute of the University of Valladolid. Thank you very much.
Thank you. Thank you very much. Last speaker is Professor Simon. Oh, sorry. Without glasses in this ophthalmology session could be difficult. Dr. Rafael Simon is the director of the Diabetes and Metabolism Research Unit at Vall d'Hebron Research Institute, head of the endocrinology department at Vall d'Hebron University Hospital, and professor of medicine and endocrinology at the Autonomous University of Barcelona. His group belongs to CIBERDEM, the Spanish network for the research of diabetes and associated metabolic diseases. He has authored over 200 articles related to diabetes, published in high-profile peer-reviewed journals, besides 12 book chapters. He has participated in 22 research projects related to diabetic retinopathy and in 28 projects related to clinical diabetes. Professor Simon is the coordinator of the European Consortium for the Early Treatment of Diabetic Retinopathy, called Eurocondor, and this international consortium was funded by the EC for developing the first clinical trial using neuroprotective agents topically administered to prevent or arrest the development of diabetic retinopathy. In addition, he is the diabetologist of Expert Committee on Diabetic Retinopathy and Vascular Diseases of the European Vision Clinical Research. He is president of the European Association for the Study of Diabetes Eye Complications and renowned worldwide for his contribution to the pathophysiology of diabetic complications, in particular diabetic retinopathy. Professor Simon has received many prestigious awards for his achievements in endocrinology and diabetes. The title of his conference is GPCR-based therapies, GLP-1 in diabetic retinopathy. Thank you.
Many thanks, Andres, for your presentation and for your kind invitation in the setting of this prestigious meeting. In the next minutes, I will try to give an overview related with GLP-1, that is the acronym of glucagon-like peptide 1, role in diabetic retinopathy. As Jose Carlos previously mentioned, it's very clear that diabetic retinopathy remains a challenge because, in fact, it's the leading cause of new cases of blindness in working age population. The question is what we are doing for treat diabetic retinopathy. We are talking to our patients that be careful with the blood sugar and with blood pressure, but we are doing nothing till very, very advanced stage. Nothing I refer specifically to address it to the eye. The very, very late stage in which we are recommending laser photocoagulation, intravitreal injections, or even vitreoretinal surgery. As you know, all these procedures are expensive, requires a retinal specialist, and also are associated with significant side effects. However, in recent years, an emerging concept is important to stress. That is that before we could be able to detect any microvascular disease in the ophthalmoscopic examination, in the retina already exists the neurodegeneration that Professor Pastor emphasized previously. And the question is that this neurodegeneration not only antedates, but also participates in this microangiopathy, correct? We have already convinced of this 10 years ago because we had the opportunity to compare the retinas from cadaveric eyes in age-mature diabetic and non-diabetic with that we have sure that the year previous death, these patients present no microabnormalities in the fundoscopic examination. And you can see here the two hallmarks of neurodegeneration, the apoptosis and glial activation that occurs, I insist, in patients without any microangiopathy the year before death. More recently, we have characterized a model to test drugs, and this model is the DBDB mouse, and as you can see here, reproduce very well what occurs in humans. Here you can see in green the glial activation, and here you can see the apoptosis, and here in the transmission electron microscopy, the strikingly apoptosis of the nuclei in the photoreceptor layer. But what is the mechanism linking neurodegeneration and microangiopathy? It's a very complex mechanism, and I'm afraid that I will have not enough time today to go in detail, but please go to this review that apart from me, I have had very proud to collaborate with these two top researchers, Alan Steed from UK and Tom Garnett from US, from the University of Michigan, in this review. And only to stress that we all produce in the retina neuroprotective factors that are downregulated by diabetes. And these neuroprotective factors are somatostatin, cortistatin, and GLP-1, among others. All of them are G protein coupled receptors proteins. So this is linked very well with the title of the conference. And we have found that when we administer topically these compounds, they prevent neurodegeneration of the neuroretinal cells. But most important, when we are enhancing GLP-1 by delivering GLP-1 or GLP-1 analogs, as we present after, or by indirectly inhibiting GLP-1 degradation by administering DPP-4 inhibitors, we not only prevent neurodegeneration, but also prevent the vascular leakage, thus demonstrating that when we are preventing neurodegeneration, we are being effective in preventing early microvascular abnormalities. Therefore, when we administer GLP-1, we are targeting the neurovascular unit impairment, and here you can see the neurovascular unit that is composed by vascular cells, endothelium, pericytes, basement membrane, but also extravascular cells, neurons, and glial cells. And glial cells are very, very, very important because when they are inflamed or activated, glial activation produce an alteration in this neurovascular unit that favors the leakage of albumin in the extravascular space, or what is the vascular leakage. And therefore, there is a disruption of the inner blood retinal barrier. So, and this is very important, and here is my friend Tom Garnett, that previously collaborated with him for a long term, is the position statement of the American Diabetes Association that is diabetic retinopathy is not defined merely with microvascular disease. Diabetic retinopathy is a highly specific neurovascular complication. Another thing is the route of administration. As has been previously mentioned, one of the routes of administration is the eye drops, the topical administration. Why? Because there are a lot of literature demonstrating that really the drugs reach the retina at pharmacological levels. In addition, is easy application, could be self-administered, and in addition, minimize the potential systemic adverse effects. And here you can see the rationale why we use GLP-1 receptor agonists for the treatment of diabetic retinopathy. First, because this receptor has been found in central nervous systems, and because the retina is ontogenetically derived from the brain, it's logical that these receptors exist in the retina. And also there is ongoing pilot studies in neurodegenerative disease using GLP-1 with effective results. But we have to demonstrate this. And moving on rapidly to the results, here you can see the design. We have used, for comparison, 8 diabetic donors and non-diabetic donors, the retinas from these patients, and also an experimental model using the DBDB mice, as previously mentioned, treated with vehicle or with several analogs of GLP-1 or native GLP-1 during 2 weeks. And here you can see, when we are analyzing the human retinas, the abundance of GLP-1 receptors that exist in all your retinas, mainly in retinal pigment epithelia, and this is located mainly in ganglion cell layer. Look at this that this expression is even higher than obtained in the bowel and the liver. We have also demonstrated that this receptor exists in the mouse model that we used. And what about GLP-1 as a protein itself? GLP-1 is this expression much lower than in its receptor, but there is a deficit in diabetic retina. And very curiously, the location is the same that the receptor. So we have, although this a small expression, there is a paracrine or autocrine action because all these locally peptides synthesized behind the receptor is very important in human disease and physiopathology. Regarding the interventional study, you can see that GLP-1 administered by eye drops is unable to produce any change in blood glucose levels. So all the changes that you will see now are provoked by GLP-1 and its signaling by GLP-1 receptor, but improving blood glucose levels. This is very important. And for the reason we also, in this experimental model, we are emphasizing the topical administration. Here you can see that the kinetics demonstrating that reaches the retina very rapidly. And here you can see the results. This is the vehicle, the sham, and here you can see the effects of native GLP-1 and all the analogs, thus preventing glial activation. I think that these images are self-explanatory. Here you can see the prevention obtained in apoptosis. All these anatomical changes were run in parallel with a significant improvement in electroretinography performed in the mice. Here you can see the proof that GLP-1 is acting by GLP-1 receptor by activating IMP sickle. Here you can see the activation. That is the first downstream mediator. Regarding the mechanism, it's important to emphasize regarding excitotoxicity. Excitotoxicity is one of the most important cause of death of neurons that is produced by an excess of glutamate. And the main molecule that produces the clearance of the extracellular glutamate is GLAST. So the higher the GLAST, the lower glutamate, and we are preventing neuron death. What happens with these retinas? The retinas, you can see the vehicle, the diabetic retina, that is an increase of glutamate, so increase of excitotoxicity on neuron death. And here you can see how GLP-1 is able to reduce the glutamate levels at the same level that the controls. Why? Because it's able to prevent the downregulation induced by diabetes of GLAST. In addition, we found that GLP-1 and GLP-1 agonists are able to regulate the pro-survival molecules in terms of increasing pro-survival molecules and decrease the pro-apoptotic signaling. And here you can see how they are able to increase phospho-AKT. We have also demonstrated that GLP-1 are able to reduce significantly the overexpression of interleukin-1 beta that occurs in diabetes, overexpression of VEGF, and also albumin leakage associated with the overexpression of these cytokines. So, but perhaps you are wondering, well, you are preventing, but what happens when you have already have a lesion? We have done this, and we have not published, even the previous results were published in diabetes, but this is not unpublished results in which we use mice of 21 weeks when already exists the glial activation, when already exists the apoptosis. What happened when we administered GLP-1? Would be GLP-1 able to regress or arrest progression? Because we were talking just now for prevention. Now we are for treating, right? you can see in this case, we have only used GLP-1 native, instead 2 weeks, 3 weeks, and of course, the mice were older. And rapidly, we proved that GLP-1 was able to regress glial activation, to regress apoptosis, to normalize LRG, and more importantly, to regress vascular leakage. So it was really amazing. We are now investigating the molecular mechanisms, but we have confirmed at these older mice that the downregulation of BGF also exists, that exists an inhibition of the upregulation induced by diabetes of NF-kappa beta, as you can see here. And this is mediated by a tremendous increase of phospho-MPK, that, as you know, is an inhibitor of NF-kappa beta. Here you can see how GLP-1 is able to impede the nuclear translocation on FNF-kappa beta, and all the cytokines downstream on F-kappa beta, such interleukin-1 beta, interleukin-6, TNF-alpha, are tremendously decreased. And finally, we have also confirmed at this age that GLP-1 is able to increase phospho-AKT, as you can see here, and also BCL, and also beta-catenin, that is a pro-survival factor. So in summary, this is my last slide. GLP-1 receptor activation produce neuroprotection, but also microvascular protection, and I think that could be emphasized as a new target for initiating clinical trials in early stages of diabetic retinopathy by route of topical administration. And that's all, and thank you for your attention.
Thank you, Rafa. I invite the speakers to the stage. We have around 7 minutes for discussion, so we can give priority to the questions coming from the floor. Any questions?
I have a comment.
Okay.
So it was very clear your explanation regarding the product that targeted for ankyrin. And ankyrin is a very interesting thing because we have found that ankyrin is downregulated in the retina of human diabetic in a proteomic analysis that we are now trying to publish very quickly. And my question is that apart from the antivascular effects that you have found, it's possible that you have also found a very important neuroprotective effect by using your drug. So my question or my offer is that we can collaborate in this issue.
Yes, of course. As you know, the NARP is a key regulator, and we suppose that we are starting the development in M&D because it's a huge market, and we work in a pharmaceutical company, but we are interested to test the NARP sRNA in other retinal diseases like diabetic retinopathy, and also we are analyzing the effect of the NARP in animal models of pigmentary retinopathy. Also, this is a rare disease with mutation in the photoreceptor, and the patient suffer vision loss in a very short time. And of course, we are open to test NARP in other animal models. The problem that we have in the retina is to find a good animal model, because you know the animal model in the case of eye diseases is quite difficult to reproduce the pathology, the human pathology, and this is the more problem. It's the only manner to test, or the only that you have clear evidence that your product works is in human, because the animal models are not very effective to reproduce the disease.
Okay, thanks. My question for you was quite related to the previous comment, is that you, in a certain way you are a pioneer developing these oligonucleotides for these indications. So, and we are talking about translational medicine, which has been the main difficulty in translating your preclinical data to clinical. That means dose, that means safety, that means efficacy. What have you learned in your developments that you can?
This is one of the more important points, the translation medicine between the preclinical and the clinic and the human. I've previously mentioned the animals model in ophthalmology are not very. They are not a very good animal models to reproduce, and the most important for us is to work in rabbits or in animals that the volume of the eye is similar to the volume of the eye in human, to translate as much as possible the pharmacokinetic and the pharmacodistribution of the drug, because we have a very specific technique to measure or to detect the siRNA that are around the tissue and work into the tissue, because the siRNA need to enter into the cells to produce the silencing, and we have a very specific technique to demonstrate that the products are around the tissue or into the cells working with HPLC complicated technique. And we demonstrated the pharmacodistribution in retina, for example, in big animals. The side of the eye, in our experience, the side of the eye is important, because the translation for the rat or mouse to the human is more complicated, and we prefer to work in animals more similar the volume that in human.
Okay, thanks. I think that, okay.
I have a comment about the size of the eye, and rabbits are very good to be eaten with rice, but they are far away from the human eye. So this is why we transfer beginning to the pig. The pig eye is quite similar in many structure, including the retina. But you have another option. This is why we are now dealing with cell culture from human origin, neuroculture of the human neuroretina, and also we can also cultivate cells from patients with diseases. So we can analyze a lot of things, avoiding the use of animals. You know that is a strong movement all over the world against the use of animals. And so there are some possibilities beside the use of animals. Obviously, primates are they're quite similar, but are impossible to afford. Yeah, I know.
I think we have a question from the floor.
Yes. Just a question for both of you, because it's directly linked to what you commented. On one side, you comment that rabbits could be a nice model because of the size also of the animal, and you commented mainly things on topical delivery. And on the other side, you commented that it's very interesting to explore new routes of administration. So you comment on the cell, but I'm on the drug delivery side, and we are working on intravitreal or other routes of administration. And the difficulty we have is that the animal model we can use, for example, Dr. Simon models, they use very small animals, so we cannot extrapolate in terms of how the drug delivery system could work on these animals. So do you have any suggestion or opinion on that?
Well, I think my suggestion is very clear, and so if you can work with pigs, it's fine, because the side of the eyeball is quite similar and the structure, and in the physiology as well. The problem with the rabbits is it's only, in my opinion, is useful just for safe tests or things like that, because the behavior of the retina is completely different, the aqueous humor, even the lens, and so on. So because we can afford primates, we use pigs, but we try to transfer to cell and tissue cultures. I think it's a good strategy. Of course, for delivering it's impossible, because you need the orbit and the surrounding tissues of the eyeball, but pigs is my option.
Yeah. I agree with Jose Carlos. The question is not restricted to ophthalmology. The question is restricted to other areas. We are curing diabetes in the mouse every week. Okay, and it's a natural report. Okay, so it's not a problem only for the eye. It's a general problem of the rodents and so on. And the question regarding the toxicity, I absolutely agree. So the rabbit is a model that is for toxicity, for preclinical. But I insist that the mouse model to performing proof of concept is absolutely okay. And then perhaps it's better to go to the clinical trial because the money that we have to invest in pigs also very, very expensive, and then is more or less the same.
I think our time is up, but in any case, I wish to thank to the speakers for their wonderful talks and contributions, and to the audience for attending this first ophthalmological session in BIOSPAIN. Thank you very much indeed.